Next, we'll be having Miss Pousali Chatterjee from West Bengal State University, who will be attesting to the role of microbiology and sustainable development. Once again, if the audience has any queries, they can type it in the chat box and we'll be addressing it at the Q&A session at the end. Over to you.
Yeah, hello, I'm audible. Sonali, my voice is clear?
Yes, you're audible.
Okay, okay. Thank you. So, thank you everyone for participating in this BioCon. And I am working here as a HR manager as a TBS, and it's a great pleasure to be the part of this BioCon and as a speaker for this particular session. And it was a great talk by Nikita. Thank you so much for your great presentation. So, I will start sharing my screen. Okay, is it visible clearly, Sonali?
Yes, it's visible. Can you please put it on slideshare mode? Yeah, cool.
Yeah, yeah. Okay. Okay. So I'll start right now. Yeah, just a moment. Um, oh, sorry. Yeah, okay. Okay. So mostly my talk will be related to the agricultural microbiology. I'll be mostly focusing on the agricultural part because when we are talking about sustainable development and our environment, then we also need to think about food, like how we can get our food, because the population is blooming every day, and there is a huge need of food that need to be fulfilled by the farmers, and we need to eat, right? So if we need to eat, then agriculture is very much vital. But on the other hand, we cannot continue use the chemical fertilizers, because those chemical fertilizers will definitely help us to increase our overall yield and plant growth. But on the other hand, those are not a eco-friendly approach. So instead of those chemical fertilizers, how we can use microorganism, and how these microorganisms can actually increase the overall production, crop yield, and fulfill our need in the upcoming future. That will be the main focus of this discussion. So basically, if we look into the soil, then even a teaspoon of soil contains billions of bacteria, and which are actually greater than the overall population of human on this earth. So that's how the large population of microbes is. And how scientists in the modern agricultural trend, the scientists are mostly focusing that how strategically we can use microorganism to fulfill our needs of food in the future. Because in the soil, if we take a teaspoon of soil, there will be billions of bacteria, but not all the bacteria are beneficial. There should be a balance between both harmful and beneficial bacteria in our soil. So how we can use strategically those beneficial bacteria, how we can identify those strains of beneficial bacteria, and how can we strategically use them, and why we are calling them beneficial, what type of activities they are actually carrying out inside the soil or in our entire ecosystem, that need to be understand very first. So microbes actually responsible to feed the entire world, and that's why we are focusing here on the sustainable agriculture and how we can use those microbes to feed the entire world. So as I mentioned, that a teaspoon of soil contain billions of bacteria, but not all are beneficial. So which bacteria are basically beneficial? So if we look into the evolution of this art, uh, I don't know that it's how many of you are really interested to know the evolution of eukaryotic system on this earth. But eukaryotes were not evolved in this earth without the presence of oxygen. And the initial earth was oxygenless. There was no oxygen even around 3.7 billion years ago, and there was no eukaryotic system on this earth. But around 3.7 billions ago, first the prokaryotic cells, the bacteria, they started to utilizing this H2S. Here it is H2O, but they initially started to utilize the H2S as their electron donor because they started to carry out the photosynthesis. Here you can see the photosynthesis, the entire diagram of photosynthesis, because we know there will be electron donor. Initially it was H2S, and if it is H2S, then it will not produce any oxygen. That's why there was no oxygen in the primitive earth condition. But the bacteria, which is named as cyanobacteria or the blue green algae, the cyanobacteria or the blue green algae, which is mostly found in the marine ecosystem, they started to use this, or they developed the biochemical pathway where instead of H2S, they started using the H2O as their electron donor. So when H2O is the electron donor in the photosynthesis system, it will produce oxygen as a byproduct, or it will release the oxygen into the environment. That's how the initial oxygen entered into the ecosystem, and gradually the oxygen concentration increased in our ecosystem, and that oxygen level or higher amount of oxygen supported the evolution of the eukaryotic cells. And the cyanobacteria started carrying out this photosynthesis mechanism around near 2.8 billion years ago. So for the development of the eukaryotic life on this earth, or for the evolution of eukaryotes, bacteria are responsible, mostly cyanobacteria. Now cyanobacteria is not only able to carry out photosynthesis, but they also able to carry out the nitrogen fixation as well. Now we all know that in our atmosphere, in our environment, that is a very huge amount of nitrogen gas is present, but we and even plants cannot able to use this nitrogen gas directly, because nitrogen gas we cannot use directly, it's not the usable form. So who can make it in a usable form for plants and for us, because we are dependent on plants for our food, and plants are dependent on the usable form of nitrogen. So who are responsible for carry out this nitrogen fixation? Several bacteria are responsible to carry out this nitrogen fixation, and cyanobacteria is one of them. Here we are talking about cyanobacteria because cyanobacteria is the free living bacteria, or blue green algae. Basically, they carry out they carry out photosynthesis, and on the other hand, they carry out the nitrogen fixation as well. So this is the overall like most simple diagram of the nitrogen cycle. And here we can see most of the part, or almost every part of nitrogen cycle is carried out by microbes, by bacteria. Most important part is the nitrogen fixation, that is carried out by the nitrogen fixing bacteria. We call them as diazotrophs. Diazotrophs, one example of diazotroph just now we discussed is cyanobacteria, and there are a lot more examples. And there are different type of nitrogen fixing bacteria. We have both symbiotic and free living. Cyanobacteria is a free living bacteria, and other examples are also there. We will come to that later. So the nitrogen fixation is basically when the environmental nitrogen gas is converted to a more usable form, either ammonium ion, or it later nitrifying bacteria can convert it to nitrite and nitrate also. And this nitrite or ammonium ion can be directly uptaken by the plant. If you see into this diagram, the nitrogen fixing bacteria convert the nitrogen gas into ammonia or ammonium ion, and then it converted to nitrate. So almost every step is carried out by bacteria, and plant can uptake through its root system, it can uptake only either nitrate or ammonia. They cannot uptake directly the nitrogen gas. So it is the bacteria that making it the nitrogen into a more usable form, and that's how plants are uptaking this nitrate and ammonia from the soil. And that's how the soil quality, the quality of the soil, the fertility of the soil, the structure and the texture of the soil is also dependent on this type of bacteria which are residing in the soil. So the soil structure will be completely disrupted if this beneficial bacteria is not present in the soil. If bacteria is not carrying out the nitrogen cycle, we can say that indirectly we will not be able to survive on this earth. Eukaryotes will not be able to survive. We are completely dependent on bacteria. So that's how plant uptake the either nitrate or ammonium ion directly from the soil, and later they assimilate those things into more usable form, like they carry out the amino acid synthesis. That is a different biochemical pathway that carried out inside the plant. Now, if we focus on nitrogen fixing bacteria, there are two type of nitrogen fixing bacteria: the free living bacteria and the symbiotic bacteria. Now, the free living bacteria are those, one example is basically just now we discussed about cyanobacteria, and there are several cyanobacteria carry out photosynthesis as well. There are certain non-photosynthetic and photosynthetic bacteria, both facultative and aerobic are there. And in case of symbiotic nitrogen fixing bacteria, they mostly form the root nodule. Root nodule is formed, which is the site of the infection of the plant roots. But this infection is beneficial because of this infection, or at the site of this infection, this rhizobacteria carry out they carry out the nitrogen fixation. So they allow the plant to uptake the usable form of nitrogen from the site of the infection. So both host gene and both the symbiont genes are required for the formation of the root nodule. It's also a very elaborate biochemical process, and in this case, plant basically secrete certain root exudate into the soil, like they secrete, for example, flavonoids and other root exudates that attract the bacteria. And rhizobacteria can sense that, and then with the help of the gene expression, they start the infection thread formation inside the root hair cells of the host plant, and the root hair curling or the infection actually formation occurs. And later on, inside the infection thread, they start to, or inside the root nodules, basically they start the nitrogen fixation process. So though it is infection, but still plants are getting benefits from it. Now, why we are talking about so much about rhizobacteria? Because this PGPR is playing a vital role in agriculture these days. PGPR means plant growth promoting rhizobacteria. In the soil, there are several bacteria in the rhizosphere region of the soil. There are the surface of the root hair. There are several bacteria are there, but only 2 to 5% bacteria are actually plant growth promoting rhizobacteria. And these days, there are a lot of research papers are there, and lot of research is also going on in this area, that how we can use this PGPR as a very strategic approach for the sustainable crop production. So basically, why we are focusing on PGPR? Because they either directly or indirectly can increase or promote the plant growth. As you can see, there are several functions, both indirect and direct way are there by the carry out by this PGPR bacteria. So they enhance the phytohormone production, they fix the nitrogen, they help the plants to stay away from other pathogenic infections, they help with the abiotic stress tolerance. So there are certain types of mechanisms are carried out by this PGPR. By the way, they can directly or indirectly help the plant and the overall plant growth and crop production. So basically, They enhance the plant growth and nutritional quality of the plant, disease suppression, and as I mentioned, abiotic stress tolerance, biomass biofuel production, and bioremediation of contaminated and degraded lands. So basically, why we can use, because we can replace the chemical fertilizers with the help of the PGPR. We can replace the chemical fertilizers, and chemical fertilizers always degrade the soil quality. They always disrupt the balance of the beneficial bacteria, and they ultimately decrease the overall crop yield and production and quality of the crop, within a long time span. So that's why biofertilizers are very much eco-friendly approach. But one problem with PGPR as a biofertilizer is that the if we use it conventionally, then sometimes we cannot get the maximum benefit out of it. So there is a recent trending in the science, in the agricultural science. If we could develop a nano biofertilizer, if we formulate some nanoparticle with PGPR together, that could be a very favorable strategy to manage the plant growth and productivity. Because nano fertilizers or the nanoparticle, like there are several nanoparticles already being synthesized, and even in certain countries like USA and in some other countries, these are already applied to the field applications. These are already being used in the field applications, and there are a lot of good results, and has been shown through these applications. And that's why it's a trending in part of, or trending topic in the agricultural in microbiology and agricultural topic these days. If we could develop certain nanoparticles, like for example silver nanoparticle, gold nanoparticles, these are already being synthesized, and if we could load this PGPR with this nanomaterial, and then we can use it as a nano biofertilizer, then that would be a very good eco-friendly approach. We can increase the overall crop production, and at the same time it will be a very eco-friendly way to sustain our ecosystem, to preserve our ecosystem, and on the other hand we can fulfill the demand of the food in the upcoming future as well. So that is the that is the main concept or topic of my discussion was today. So basically I'd like to conclude with that we need to eat, and plants are the base of our terrestrial ecosystem, but their growth, their productivity is determined by this diverse group of microorganisms that are present in the soil. So if we could use this, and this global warming, this pollution, these are real concerns, and if we could use this PGPR in a more strategic way, then as a stress mitigating agent, as a growth enhancing agent, then we could solve this insistent problem with our food and crop sustainability, and with the help of the microbiology. So yes. Okay, so am I audible right now?
Yeah, thank you so much for Ms. Pousali for the insightful session. I think we have a question.
Yes.
Okay, so there's a question from Albert. Are there any Indian companies that are working on nano biofertilizers, or are all over the world? Are there any companies that have started producing it on a commercial scale?
Yeah, in USA, as per my knowledge, and I have found in some research papers also, that in USA, I don't know exact company name, to be honest, but in USA, it's being used in the field applications according to some research papers. In Japan also, I guess, but in India it's still in the research level, I guess. Like it's still in the research level, and there are so many researches need to be carried out in this particular area of especially in the nano biotechnology area, because PGPR is already being used, but nano formulation approach is still under the research, and it's a trending part in agricultural microbiology domain.
Okay, thank you so much. I hope that's answered. Yeah. your question, Albert. Now, there's another question from Bhupesh. How much our reliance on chemical fertilizers can be reduced by microbes? I mean, plants not only need nitrogen, they also need other macro and micronutrients. Do we have some data?
Yes, as I mentioned that if you look into this slide, that not only I focused on nitrogen fixation because it's a huge important part, and we have a very good source of nitrogen gas in our atmosphere, but other activities are indirect and both direct way are carried out by the PGPR, like phosphorus solubilization, siderophore chelating, like they are the siderophores are iron chelating agents. So those type of things are also directly produced or promoted by this PGPR. So it's not only about nitrogen, but the other activities are, I have not focused in this area in detail because it will take a lot of time. But other nutrients uptake also be increased with the application of the PGPR. So other nutrient uptake, like overall nutrient uptake will be increased with the application of the PGPR in the rhizosphere area.
Okay, I hope that's answered your question, Bhupesh. Does anyone else have any questions? You can unmute and ask as well. Okay, I think that's about it. Thank you so much, Pousali. It was very insightful and interesting.
Thank you so much for this opportunity.